Hello, I am back. So, full disclosure, I was going to do this whole thing as one, but literally, in the last one, I just got to the point where I needed a break, so that's why I stopped it. And I came back and looked at this, and actually, I really don't have that much left, so I probably could have powered through and finished, but oh well, you get it either way, right? So, this one's probably going to be quite a bit shorter than the other video. All right. So what we've done so far is we've talked about amino acids individually, some what they are, some of their properties, how they change according to changing pH. What happens when you link them together, right? And this is going to be the basis for proteins. Now, before you can get to proteins, though, you got to go through the peptide stage. Peptides are formed when you form a bond between two or more amino acids. It's not really a protein. There's not a magic number, but it's not really a protein until you get at least like probably several hundred of them together, right? So we can form peptides, and there's a lot of really interesting small peptides, such as di and tripeptides, and similarly sized ones. All right. How do you actually make a peptide? Well, to do this, you make something called a peptide bond. And I'm sure you've probably heard of the phrase peptide bond for eons and eons, probably even since high school biology classes, right? So what the heck is a peptide bond? Well, a peptide bond is formed when you have the amino terminus of one amino acid reacting with the carboxy terminus of another, all right? So let me show you how this works. And if you had me for organic chemistry, you might remember that we went through carbonyl chemistry, and there were a lot of carbonyl reactions that we had, and depending on what you were doing there, required certain conditions. Like sometimes we needed a catalyst, sometimes we didn't, etc. Now, these are going to be basically applied versions of those. Now, they're going to work a little bit differently in biological systems, right? For example, we're not going to have sulfuric acid in the body, hopefully not anyway, as a catalyst to help, get any of these reactions to go along. But you do have other things that can help these formations, such as different enzymes, right? And so what I'm going to do is, since there's so many varied enzymatic mechanisms for sort of stimulating the formation of a peptide bond, I'm going to give you the very generic one that sort of shows you the sum total of what's happening. All right. So let's say we have this. Let's say we have an amino acid, and I'm just going to draw the carboxy terminus part of it. And so, again, this can happen sometimes when this is protonated, sometimes when it's deprotonated. I'm going to draw you the most generic, simplest form to understand, and that's going to be in the fully protonated version. All right? And I'm going to use just R here to represent the rest of the amino acid. All right, so we have a carboxy terminus, and then we also have an N terminus. And again, for simplicity's sake, I'm going to show you the fully protonated version. All right, and then R, again, represents the rest of that amino acid. So basically, here's what happens. It's a very simple general mechanism, actually. What happens is the nitrogen can attack the carbon on your carboxy terminus, and OH minus can be given off. Now, OH, actually, sorry. I made a mistake. Let me fix this mistake. You can't do this with NH3+. It's got to be NH2. Sorry about that. Because otherwise, nitrogen would have 5 bonds, and we can't have that. So, sorry. All right, so nitrogen can lead in with these electrons, attack your carbon, knock off OH, and it gives you this. And then that OH minus can come back and grab a proton from there. And I'll just draw this way up here. That shouldn't be a charge right there. That should be a prime. Sorry about that. And that gives you your peptide bond, which is right there, right? And you see you also end up giving off a water molecule in the process. Now, real life, if you put, just a carboxy and an amine together like this, it's not going to react in this fashion, but this is somehow catalyzed. And so basically, usually this is going to be an enzymatically catalyzed reaction, which means one of these will bind to an enzyme and there'll be a shifting of stuff where you get attacked. So there's too many different ways for it to happen. So this is just sort of a good all-purpose way of remembering how you form that peptide bond. All right? Peptide bond is really just analogous. It's a synonym for what we call an amide bond. Because we have actually now formed an amide, right? Remember when we looked over the functional groups? Carbonyl with a nitrogen attached to your carbonyl carbon, that's an amide, right? So this actually forms a secondary amide in this example. I hate it when I keep doing that. All right, so why is the peptide bond so magical? Peptide bonds are essential. They're crucially essential in allowing proteins to have stability and to have direction as far as getting folded in a particular arrangement and then staying like that and making them harder to basically mess up structurally, right? The reason for that is right here. The peptide bond is unable to undergo free rotation. That's very important. Remember, free rotation in molecules. Like, if you have a carbon single bonded to something. Hey, let's say it's a nitrogen, in fact, right? Let's say it was, CH3NH2, right? Anytime you have a single bond, you can freely rotate around it, just like propellers spinning on a helicopter, right? And in OChem, we saw that, this movement, this spinning around a single bond, that can enable molecules to do certain things when certain groups were in certain positions, right? And so there were reactions that could occur in one form or one point of the rotation and couldn't in another, right? But in peptide bonds, you don't get this free rotation. And this might surprise you because, hey, that's a carbon-nitrogen single bond, just like we saw in this example, right? But in this one, we get no free rotation. You don't spin around that. Why would that be? Give you a second to think, ponder about this. Think of some basic organic chemistry principles, and that will actually tell you the answer. Notice I'm drawing in the electrons that are found here. All right? All right. If you look here, remember, if you have available unshared electrons on heteroatoms like nitrogen and oxygen, And also, if you have pi electrons, you can have, a lot of times, resonance. And that's what we have here. This is one resonance structure of a peptide bond, and it's the most obvious one, right? Probably the most stable form. But hypothetically, you could move this pair of electrons into a double bond with carbon. That would give carbon 5 bonds, but it could move a pair of pi electrons up here. And if it did that, it would form this resonance structure, right? And guess what? In this resonance structure, we actually see a carbon-nitrogen double bond. And what do we remember about rotation around double bonds? It doesn't happen, right? When you have a double bond, those atoms are locked into place. They don't move. They don't rotate around the bond, right? So that keeps them there. Remember, if you have resonance structures, really, neither one is the full truth. The truth lies somewhere sort of in the middle. So it's kind of a 1.5 bond between those two atoms here. If we were to draw the resonance hybrid, it looks something like this. Partial positive on that one, partial negative there. Right? But since this nitrogen and this carbon have to have the ability to have this what we call partial double bond character, that partial double bond character allows us to say that there's no free rotation. And so that means those atoms are locked into place, and that means if you have a bunch of amino acids linked together, they're going to be in a very set order and a very set orientation that allows the protein to fold up, assimilate, and form binding sites in a very regular way that doesn't change, right? It allows them to do their job as a result, right? So that's why the peptide bond is so important. It gives these amino acid chains partial double bond character that allows them stability and consistency. All right. Peptide group is planar. All three of those atoms in the peptide bond, the carbon, the oxygen, and the nitrogen, because they have this partial double bond character, all of them are sp2 hybridized. And if you'll remember, sp2 hybridized atoms have trigonal planar geometry around them. All right. When you start putting amino acids together, right? Let's say you make a chain of 2 of them, or 3, or 4, or 500, or 1,000, or whatever. You do have to describe the sequence of amino acids in a certain type of nomenclature. So whenever you see several of these listed in a row, I thought I might have an example here, but maybe I don't. There should be a picture in your book. But they are always listed. The first one listed is always the N-terminal end. The other one is what we call the C-terminal end. Oh, and I didn't realize there was already a picture of the resonance here, but you saw it in my drawing, and you can see it here. All right? So, like I said, there's all sorts of interesting and useful peptides out there that are not proteins, right? They're not big enough for proteins, but they have other functions. Here's one you've probably heard of. Here's one called aspartame, right? Where have you heard of aspartame? Obviously, if you can read the slide, you can say sweetener, used as a sweetener. But yeah, you guys have probably, if you've ever had a Diet Coke, right? If you've used certain types of sweeteners in your coffee, right? A lot of those are based on aspartame. And aspartame is actually not a sugar derivative. It is a peptide. It's a dipeptide derivative. It's formed between aspartic acid, and then phenylalanine, a phenylalanine that has been sort of modified to have a methyl ester tail instead, right? So, I don't know if I have phenylalanine on here. Give me 1 second. Hold on 1 second. Sorry, I got distracted by my dog over here. Sorry. Dogs do that. What I'm going to do here, real quick, is I am going to look up the structure. Of aspartame and just sort of highlight. There we go. So if you look at aspartame here, basically on the left side we have aspartic acid, and then it's been linked to a phenyl ester of phenylalanine. So in the middle, right, you see a peptide bond, C double bond O with a single bond to N, and phenylalanine has been modified to have an ester group on it. So, like we said earlier, sometimes these 20 essential amino acids can be modified a little bit before their use. Okay. So yeah, and it just so happens that this particular arrangement, when this binds to a lot of the receptors for these peptides in the body and the taste buds, it will give a sweet taste. I actually personally do not like aspartame. I hate the taste of it. To me, it tastes kind of bitter, but to a lot of other people, it tastes sweet. So again, it kind of goes to show that the formations of different receptor sites can interact differently with incoming substrates. And so we'll talk a little bit more about that once we get into proteins and enzymes. You may have heard some concerns about these. There are some legitimate health concerns for people who have something called phenylketonuria, or PKU. All right. So basically, what happens there is there's a deficiency. A lot of these metabolic diseases like this will result in, will result from enzyme deficiencies, right? So basically, how it works here is that let's say you have phenylalanine. Well, just say PHE phenylalanine. All right. There's a couple of different fates of phenylalanine once it gets into the body, right? It can be converted, and some of it can be converted into tyrosine, which then can enter various other metabolic pathways somehow, or it can be converted to something called pyruvate. All right, and then phenylpyruvate is then converted into phenylacetate, which then gets converted into, excuse me, sorry, I skipped one. Oh, son of a nutcracker. Hold on. Oh, good Lord. phenolactate, and then that can finally be converted into phenylacetate. All right, so all sorts of conversions that can happen here to various species, and sometimes some of these various intermediates will be sort of inserted into certain metabolic pathways. All right, other ones can literally be transformed into species that will be flushed from the body, waste products, right? So it can't be flushed in one form, but it could be flushed in another form. All right. Well, phenylalanine, when it's converted into either tyrosine or phenylpyruvate, both of those processes are enzyme dependent. Conversion into tyrosine is dependent on an enzyme called phenylalanine hydroxylase. You'll notice a lot of our enzymes will end with the suffix ase, and then the rest of it sort of can describe what's going to actually occur. So basically, when you have phenylalanine hydroxylase, that basically means it's an enzyme that catalyzes the addition of a hydroxy group onto a phenylalanine. So if you compare the structures of phenylalanine and tyrosine, you'll see they're similar, but tyrosine has no H group. All right. The conversion to phenylpyruvate is catalyzed by something called transaminase. That's supposed to be an M, by the way. Transaminase. A transaminase is something that can transfer an amino group from one thing to another, right? So, this is what normally happens to phenylalanine in the body. However, to somebody who has the condition PKU, they have a deficiency in phenylalanine hydroxylase. And that has pretty dramatic effects, right? Basically, if you're deficient in the enzyme phenylalanine hydroxylase, that means that a certain amount of phenylalanine that usually gets turned into tyrosine can't be converted, right? You can't form that tyrosine. So, one thing is that can lead to the tyrosine deficiencies elsewhere, but the more important thing that happens is that means you end up making more of this than you should. And that's really, really, really bad, right? Because one thing this does is phenylpyruvate will, if you have too much of it, it will actually inhibit the conversion of pyruvate to something called acetyl-CoA. And you see this conversion several places in several metabolic pathways. It's a very important reaction. Okay, but we have these things called inhibitors, right? And if you have an excess of something, a lot of times in the body, it will interact within a pathway and sort of say, hey, you got too much of this, you don't need to make more. So what happens is excess phenylpyruvate will sort of jump in here, and its presence will inhibit this conversion. And that's has a lot of very severe effects. People with PKU, because they have this accumulation of phenylpyruvate, they will undergo some pretty severe symptoms. Most notably, very, very impaired cognitive development. So if this is not caught early, that can have a pretty dramatic effect. You have PKU and you're eating phenylalanine containing products. You're going to be very, very cognitively impaired, and that can eventually be toxic as well. So this actually is a controllable condition, which is nice, because you can sort of, if you know you have this deficiency, you can adjust your diet to make sure that you don't consume as high, phenylalanine in as high levels as you might normally do, right? So if you look on a lot of foods that contain phenylalanine, you'll actually see somewhere on the packaging that will say something as a warning to phenylketonuria. Hey, this has phenylalanine in it, right? So if you have PKU, probably don't want to eat very much of that, if any at all. All right. Let's see. Another one. Oh, sorry. Going back to what we said about aspartame. Since aspartame has a derivative of phenylalanine, aspartame is quite dangerous for people with PKU because once aspartame is cleaved, that peptide bond is cleaved, phenylalanine methyl ester can be converted back into phenylalanine, and that can result in the same problems we just mentioned. So that would be one of the things that you would want to avoid. Let's see, a couple other ones that are interesting. Glutathione is a tripeptide, right? It's got glutamic acid, cysteine, and glycine all bonded in that order. This is an important one because this is a good scavenger of oxidizing agents. All right, and that's probably a nutritional buzzword or buzz phrase you've heard over the years. If you've ever heard radical scavenger or free radical trap, or so I'm blanking on them now. Sorry, but a lot of times, especially in what people call superfoods, they'll advertise something as a superfood because it's high in levels of certain types of compounds that will sort of scavenge or eat up free radicals in the body. And these free radicals, they're harmful in biological systems because they can cause oxidation reactions, unwanted oxidation reactions in the body that can hurt various things, including nucleic acids. All right, so this is kind of nice because this particular one, if you look here, what it does is it's got a thiol group on it, or as they call it, a sulfhydryl group, because of that cysteine, right? Remember, cysteine has SH. And what happens is if you have two glutathiones present, this can actually interact with oxidizing species and sort of, in a nutshell, each of those SH groups gets oxidized instead of an important biomolecule. In other words, it kind of takes one for the team, right? So instead of, let's say, an important covalent bond in a strand of DNA being oxidized, instead, two molecules of glutathione will be oxidized. That's pretty cool because that means that those oxidizing radical species have been sort of, I guess, eaten up, for lack of a better phrasing, but without damaging your important biomolecule groups. All right? And so what this does is this will form a disulfide bond whenever you oxidize these guys. And then it doesn't hurt you at all. It doesn't hurt you at all to lose these individual glutathiones. It doesn't hurt you at all to make the oxidized version, which is GS just G. It's not a harmful compound. And you can regenerate glutathione in the body. So some of the superfoods that you may have heard of, some of those advertised will contain some higher levels of glutathione in there. Another interesting group is enkephalins. A lot of people get these mixed up with endorphins. These are similar but kind of different. These are pentapeptides, right? And there's a couple of different forms of them that are almost exactly the same, except the carboxy terminus, and one of them is a leucine and the other one is a methionine. So if you were to look at the amino acids in these enkephalin peptides, you'll see that a couple of them, tyrosine and phenylalanine especially, those have aromatic groups. And one thing to remember in biochemistry is that shape, molecular shape, stereochemistry, three-dimensional arrangements are hugely, hugely important. When we get to proteins and enzymes, right, we're going to see that the folding of all these proteins can result in what we call an active site where something can bind. And literally, the shape of that active site will almost, I don't want to use the term lock and key because that refers to something else, but kind of like a hand in a glove, right? It kind of only allows a certain fit, right? And so, having a certain amount of aromatic groups can make for a certain shape interaction between an active site and a substrate, right? So, one thing that's interesting is that, we have receptors in our body that can bind with enkephalins. And we say these are natural analgesics. An analgesic is basically sort of a pain reliever, right? So if you have a spike in enkephalins and you have these enkephalins binding to the right receptors, that can sort of cause a pain inhibition triggering, right? So you feel better. And in fact, what a lot of people, if you've ever heard somebody describe like a runner, for example, somebody who runs and they get really tired and achy and stuff, but they quote unquote break through the wall. It is possible at a certain point you get to a point to where your natural body state will trigger the release of these enkephalins, and they will bind to your receptors and make you temporarily feel a little bit better, right? They'll eliminate some of that achiness. And one thing that's kind of interesting is that, and this is not just with these, but you can see tons and tons of interactions in forensic science where drugs and pharmaceuticals of various types can bind with the same receptors that your natural molecules will bind to, and they can sort of mimic their activity. An interesting one is morphine, right? And you guys probably have all heard of morphine. Morphine is a really, really strong painkiller. They'll give it to you in the hospital if you have surgery, and you have to have it in very, very controlled doses because it's very addictive, right? Here's the structure of morphine, and you can see the structure of morphine has a number of different aromatic rings, and it just so happens that the structure of this, and here's the structure of one of the enkephalins here, when approaching an active receptor site for an enkephalin, it can sort of mimic its shape, and it fits in there pretty well, and it sort of artificially triggers the same biological effects. So when you get morphine, that's why it's causing a pain-killing effect. It's because really it's sort of mimicking what would happen with your natural analgesic here, in the enkephalins. And then related compounds. Obviously you have some other ones too that are either related or straight derivatives of morphine. Heroin, of course, is one. Fentanyl, and a lot of your oxycodone and stuff like that. To varying degrees, by sort of tweaking their structures, you can make them fit into these receptors maybe in a slightly different fashion that causes more, or in certain cases less, of an effect. You see something very similar with, I'm getting a little off topic here because this is not peptides, but if you talk about methamphetamine, right? Methamphetamine's a stimulant. It can very much mimic the activity of something called dopamine. Pretty interesting. Couple of other ones that are interesting. Oxytocin is a structure, and I don't have the structures here, but you can find them in your book. It's got a cyclic structure that is formed through a disulfide bond. And it's very similar to another peptide called vasopressin, right? Very similar in structure, except there's two minor differences. And you can see that even making minor differences in peptide sequence, identity, or stereochemistry, it can make a big difference in how they function, right? Oxytocin is a peptide that is present in a couple of different situations, especially within mothers, right? It's released to help induce labor, and also it's the peptide that helps to stimulate milk production when a mother is breastfeeding, right? Vasopressin has an entirely different function. It stimulates reabsorption of the water by the kidneys. So it goes to show just how slightly changing a sequence or a position of an amino acid within a peptide can greatly change its activity. Also, fun unrelated fact here, or sort of related but not chemically. Um, if you ever look into, if you're like an animal lover, and like you and your dog or you and your cat just really like to sort of gaze into each other's eyes, and it just makes you go happy, and it makes the animal happy, that's because they're literally, when there's a bond there, that actually stimulates a release of oxytocin, actually within each of you, to sort of give you this nice, warm, happy, loved feeling. Very much like the feeling between a mother and a baby. So there you go. If you want to ever give yourself a nice little oxytocin rush, just find your favorite pet, gaze into their eyes lovingly, and it'll make you happy. So that's how I'm going to end it, because that's a really happy way to end it. So that's the end of this one. Listen to these, make notes, correlate with the chapters in the book and the PowerPoints, and we will talk about this together as well. All right. Bye bye.